So here we have the cell membrane, and then of course outside that cell membrane you're going to have a cell wall. And that cell wall can vary depending on the microbe that we're talking about. You'll have to know the difference between a gram-positive and a gram-negative microbe. In addition to having the cell wall, some microbes have a glycocalyx. It's like a jelly layer outside the cell, a capsule if you will. Serves the cell in allowing it to stick to surfaces. Biofilms develop because of this capsule. Some microbes, like Staphylococcus, doesn't have a capsule outside your body, but once it infects you, it produces a capsule. The capsule protects it from your immune system, from your immune cells. Bacteria have flagella, not all, but many. The flagellum can either be polar, or you can have peritrichous flagellum, which means you have many over the surface of the cell. Not to be confused with the flagella that we see in prokaryotes. Different, even though their function may be similar, the structure is different. They're made up of different molecules. Other things that we'll find here, of course, are circular DNA plasmids. Some cells have one. Some cells may have many plasmids. And again, when we talk about giving a cell an advantage, think about what plasmids might do. Okay, so you might have, 1, 2, 3, 4, 5 different genes on this plasmid. One of those genes might code for a toxin or an enzyme or something that gives the cell an advantage over other cells. And maybe this is picked up, as we talked about in the genetics chapter, through sex pillars, so through conjugation. Again, the gene could give the cell extra qualities that it didn't have before. Pili, we find them extending out from the surface of the cell. And these serve to do a number of things. They can help the cell stay attached to a surface. They can help a cell move along the surface through a little twitching motion. And as we saw, they can aid in conjugation. A sex pilus can be used to transfer or transmit plasmids from one cell to another cell. Other things that we might see in there, of course, there'll be ribosomes. I say, of course, you should understand the ribosomes. This is where the amino acid gets sequenced, or the amino acid chain gets put together, I should say. At these ribosomes is where the messenger RNA hooks up, transfer RNA then delivers the amino acids to these locations. And then that's where our proteins are built, put together. So you'll have ribosomes there. And then finally, some microbes produce storage granules.
What was the outer layer called again? The pink one?
This is the glycocalyx, also known as the capsule, or a slime layer. Now the difference between a capsule and a slime layer, they're both considered glycocalyx, but the difference between a capsule and a slime layer is a capsule will be entire, will be complete around the cell, and a slime layer will be more diffuse. It won't be complete, it'll be broken up, irregular.
Did you say storage granule or storage backbone?
Storage granule. Again, you should be able to reproduce the structure of the cell wall for both a gram-positive and a gram-negative cell. Do you want me to draw that, or do I need to? So, so that would be a gram-positive cell. That would be a gram-negative cell. Our inner phospholipid bilayer, our peptidoglycan layer, our outer membrane that includes a leaflet of a phospholipid layer and a leaflet of a lipopolysaccharide. Okay, so this is our lipopolysaccharide. This is our O-specific molecule. Kind of serves as an identifier of the cell. And this is our lipid A molecule. It serves as an endotoxin, triggering an immune response when the exposure is high enough.
The tails that are the lipid A, right?
I'm sorry?
The tails are lipid A.
The-
Squares, are the squares lipid A too?
Yeah, the square and the tail together. The whole thing is the lipid A molecule.
So then the glycocalyx would be on the outside exterior of that.
Yes.
That on the ground.
Yes.
But we wouldn't draw the glycocalyx on there, on these.
Not on here, no.
Just on the cell.
Over here you would, but not over there. So, you should also be familiar with how a microbe moves by the method of run and tumble. Understand the concept of attractant or repellent and how that affects the microbe's movement, you know. If it's being attracted to something, you're going to see longer runs, less frequent tumbles. If it likes where it is, it's going to tumble more frequently because it's going to want to stay in that area. Recognize the behavior is the same for an attractant and a repellent. Often when I ask that question as an essay, the students will say that when attracted to something, it tumbles frequently and stays in an area, and when being repelled, it runs away. But that's not true. The action is the same whether it's being attracted or repelled. It wants to move away or move toward. If it wants to move, it's going to have long runs, less frequent tumbles. If it wants to stay where it is, more frequent tumbles. Okay? Some of the things that can affect the movement, molecules in the microenvironment. We talked about chemotaxis, aerotaxis, phototaxis. So things that will attract the microbe or maybe repel the microbe. Be familiar with a eukaryotic cell and how it differs from a prokaryotic cell. Okay, a eukaryotic cell then is going to have the membrane-bound nucleus, it's going to have the organelles like the mitochondria and the chloroplasts. And the fact that eukaryotic cells take things into the cell differently than prokaryotic cells. Prokaryotic cells need ion channels, protein channels, to move things across the membrane into the cell, either through the channels or through diffusion. But eukaryotic cells, they engulf their material through the process of endocytosis or phagocytosis. So be familiar with that process. And then lastly, understand the theory of endosymbiosis, where our mitochondria and chloroplasts came from. I might ask you what the theory means, or what the theory is, I should say, define the endosymbiosis theory. But then I might ask you to provide the evidence that we have for that. That, of course, are the double membranes, the fact that the inner membrane is more similar to bacteria than the outer membrane. The DNA, the fact that mitochondria and chloroplasts both have their own genetic material. And finally, the ribosomes. Mitochondria have ribosomes that are more similar to bacteria than to eukaryotic cells. And finally, and probably the nail in the coffin, is that the microbe Rickettsia, which is an intracellular obligate microbe, or obligate intracellular microbe, has genes that are similar to mitochondria. In fact, they share, I think it was 4 genes that have been sequenced that microbe and mitochondria share. That pretty much seals the deal. The mitochondria were at one time free-living microbes. Any questions about chapter 3? Chapter 4 and chapter 5, a couple of things. Both of them are dealing with prokaryotic growth. One of them is, the parameters really that enhance prokaryotic growth, and we talked about the environmental conditions that have to be established. And then chapter 5 talks about controlling microbial growth. Each of them have terms in them. I would encourage you to take the time to know the terms. Now, many of the terms you're already familiar with, you probably use, but be sure you understand the definition. It's very easy for me to give you matching, where I throw up a definition of several terms and then have you put the terms with those definitions. With regards to chapter 4, one of the things that you need to understand is the bacterial growth curve. Be able to draw the curve, be able to identify the stages of the curve and what is happening in each of those stages. And when you go to draw the curve, if I ask for it, you need to draw the curve as it's represented like this for each of the stages. I don't want to see this, because that's not the bacterial growth curve. That's just a curve.
Can I ask something?
Yes, absolutely.
Can you clarify again how the colony, when you have a colony on a plate growing compared to a broth?
Okay.
Explain that before.
So this has to do with the bacterial growth curve too. So we've got a colony growing on our plate here. Now with that colony, initially when it starts, we're assuming it starts out as a few cells, 2 or 4 or something. Those cells when you first transfer them, they're going to start out slow, the lag phase, acclimating to the environment, making the enzymes and proteins that they need to use the nutrients that are here. Then once that's done, they'll grow exponentially, logarithmically. That means that the colony, starts to increase in size during this time here. Right about up here, what we start to see is that the cells in the center now, maybe you're up here, while cells on the edge might still be down here, because what's going on is in the center, the nutrients are being depleted and the waste is accumulating. So living in the center of this colony now, being one of the firstborn, not so good because you've used up all the nutrients and you've got a lot of other cells around you. Okay, so you're probably now moving later into the growth curve, or the cells around you. Or if we were to look at, if you were to look at 1,000 cells at this point in the colony, they would probably be over here somewhere. But if we were to look at cells on the edge of the colony, they don't have the same problems. They don't have the accumulation of waste, and there's some there, but they can